Hi, Ken. It's great to be here this evening.
Firstly, and most importantly, are you a beekeeper?
I am. So I was trying to think back, and I couldn't recall if it was 2007 or 2008. But I think probably 2008 is when I started keeping bees. But I've always kept personally at a hobby scale. So I think the most colonies I've ever been at is probably about 15. Right now I have one, and it's my first top bar hive that I have. So I'm dabbling in figuring out what that looks like as a beekeeper as well.
Oh, nice.
Here's my bird that we live with, so that's nice.
You've come a long way from being a zoologist studying cats in America to being a senior scientist at Plant and Food Research in New Zealand. I was very interested in your paper that looks into how nicotine reduces the cough reflex in cats. This probably explains why I always cough for a month after giving up smoking. How did you come to live in New Zealand?
Yeah, I do have a bit of a windy path to get to New Zealand and to get to bee research in general. I started out in animal science and for my undergraduate, and growing up, you like animals and everyone's like, are you going to be a vet? And I knew I didn't want to be a vet. And I had taken like a career thing that said I would do wildlife rehabilitation would be fun, but that's not, you can't really get paid to do that for the most part. So I realized or decided I was going to be a zookeeper. So I started work as a zookeeper after uni and did some internships during uni for that. And when I moved with my partner at the time to a new area, and I just didn't transition over to that zoo very well. It just was hard to make the adjustment. So I started working as a vet nurse. And while I was there, then started doing wildlife rehabilitation on the side as a volunteer project. And at that time is when I met someone who was keeping bees. And so I'm a bit of a serial hobbyist and was like, well, that sounds cool. I'd like to do that. And I took actually a local extension program that was offered through Clemson University at the time that was just learning about basics, how to be a beekeeper. Started beekeeping then and realized that I wanted to do something about going back to school to do some sort of graduate degree, to put myself in a position to have some sort of different job and some different career, and wanted to do research. And then it simultaneously at the same time was like, bees are pretty cool. Maybe I'd like to do bee research. We moved again to Florida. And at that point, I still didn't have everything fully committed to start graduate school, but got a job in a lab as a research technician. And that was the lab that did upper airway control of cats. So it was cough and swallow. So I definitely am just one of the middle authors because I was a lowly lab grunt who was doing a lot of the technical work just to have these experiments happen. But it was work funded with a human research perspective. So thinking about Parkinson's disease and how often it's the discoordination of cough and swallow that lead to pneumonia in Parkinson's patients. And so looking at the brainstem control of those two mechanisms. And so then that's when I decided that I really actually did like doing research professionally as a career. I still really thought bees were pretty cool and was enjoying beekeeping. And so that's when I got in touch with Jamie Ellis, who's going to be at the beekeeping conference this summer as one of the keynote speakers. He's faculty at the University of Florida. So I did my master's and PhD with him in honeybees at the time. And then I finished that in 2018. And that's what I moved to New Zealand with for this position at Plant and Food Research for beekeeping as well. So it's a bit of a topsy-turvy journey to have gotten there, and comes up at the end with a pretty weird collection of things that I've done.
Mm-hmm. Sounds great getting paid to work with bees.
Yeah, it's pretty fun. It's been a good trip.
What is Plant and Food Research? I see it's a Crown Research Institute. Is this an NGO?
Yeah, not an NGO. So when I was applying from the United States, it was likened to me as something similar to maybe the United States Department of Agriculture. It's a little bit different than that because it's not fully a government agency. Like it's not in with NBIE or NPI or any of the ministries. So it's a company and operates as an independent company, but our shareholder is the government. Oh, I'm like, now I'm like, it's the Crown. It's the Crown because we're a Crown Research Institute. So it's a private company, but it's in the place of what would in other countries be kind of those government agency research organizations. And so our core business, our core focus is to provide benefit to New Zealand through the research that we do. And so there's several, maybe 9 Crown research institutes that cover a broad spectrum. So there's Scion that does forestry. There's some NIWA that people have usually heard about that has to do with water. So there's like the ocean, some ocean work, and then also some of the weather monitoring that you'd see with rain that comes up with NIWA. And then plant and food research is horticulture and arable crops for the most part, but it also has a pretty strong seafood branch. So they're out there is kind of weirdly related to the core business. And then same, I feel like honeybees, we sit kind of to the side of core business because we're not breeding cultivars of kiwi fruit, but we are providing pollination for much of the horticulture crops. So that's how we sit in with plant and food research, is through that pollination scope. But then since we're the bee team, we do a whole array of beekeeping work that most people at plant and food look at us a little bit strangely about why that matters, because they're very used to hearing about plant cultivars and plant diseases and fertilizer.
The study on shook swarming is interesting. In New Zealand, it is banned for treating or mitigating AFB, so most people don't even consider it. But you looked into it for other reasons. Does it really help to reduce other diseases and chemical residues in a hive?
Yeah, so we've done some preliminary work with shook swarming, and we're actually currently pursuing a bit more funding with MBIE to look at shook swarming, but also some other wraparound handling procedures and how those might impact colony function, particularly around deployment for pollination and what that might look like for drivers for foraging. And the preliminary work that we did, we saw reduced varroa buildup in the colonies that had been shook swarm compared to the ones that had not had the shook swarm, which makes sense because you see the break in the brood cycle. It's definitely, and it in terms of the reducing chemical buildup, if you're shook swarming onto foundation, then you don't have any of the older wax that would be sequestering agrochemicals from the environment or miticides from treatments that you've put in as well. So it's one way that could be worked in terms of reducing the pesticide residues. We didn't test that. That's still a hypothesis, but it seems like a reasonable hypothesis to make. And some other work that James Sainsbury had done several years ago looked at treating of old comb, so between the spring and between the autumn and the spring with honey supers, heating those, and then that reducing nosema pressure was the hypothesis. It wasn't tested to confirm if it was nosema or not, but they just treated it and then saw that there was, did the heat treatment and then saw that there was benefit in terms of honey production in the colonies in the following season. So if things like pathogens were a part, which is what we suspect was a part of what caused that effect, then also shaking onto these new foundations would it does put the colony in a very different physiological state. And you don't have the brood that's come in, and you don't have wax in, so they're building that up. So it comes with other energetic demands that may not prove to be worth the benefits that you see for pest reduction, for maybe honey production, but we think it might be something that could actually be quite worth all of the downsides that you see for in terms of where your honeybee colony is at for strength, still for pollination.
Mm-hmm. Yeah, it might be very interesting for hobbyists as well then. Studying bees and beekeeping is very difficult as it's such a complex system and there are so many variables. Overstocking seems to be one of them. It's much easier to say how many cows or chickens per hectare, but bees?
It's pretty tricky. They don't really care where you've put a fence line or where you say that they should be grazing out in the environment. They're definitely going to go wherever they want to. Even maybe if you've put them in the middle of an orchard that you want them to pollinate, if there's something that's in bloom and yummy just a bit outside, they're going to also fly over there. Academically, it's a pretty fun question. This is like a brain teaser. Practically, if you're trying to actually make decisions in the field about where to put your colonies, particularly around a manuka flower and what that's going to look like in terms of overstocking, it's still a bit of a hassle for everyone. But it's something that we have started looking at, we've had one master student who just completed a trial last year. It is more of a case study because we had one really heavily populated area, apiary, and then one smaller one. But we have to start somewhere, and the scale at which we can't set up with plant and food colonies only very many apiaries that are more than 180 colonies, because that's most of our research apiary. So we had one quite large apiary site that was 160, 180 colonies, and then smaller apiary sites that were about 8 colonies each, and looked at the signs of physiological stress that those colonies may or may not have been exhibiting as a first shot to figure out, are these indicators that we could look at to determine if there's overstocking stress that's experienced by the hives, and if that's something we could use to practically assess overstocking. But we're still, just finished up the sampling, but we're still in the process of organizing all that data to have a clear, great answer from this part of it to carry forward into other research trials, and then also probably be pursuing some more research funding, possibly through Marsden next year, to continue that project on.
Many of us become beekeepers to save the bees, only later realizing that Apis mellifera is only one of 20,000 bees. And becoming a beekeeper to save the bees was like becoming a chicken farmer to save endangered birds. Does beekeeping negatively impact our native bees?
That's a really fun way to think about it, with the chicken farming and endangered birds. Yeah, it's harder to say definitively whether it's just neutral, whereas like beekeeping is great and fine, but you're not saving the bees, because particularly in places like New Zealand or even the United States, where I'm from, honeybees aren't native to those areas. So in terms of environmental preservation of a species, you're not doing that in New Zealand or in the United States as a beekeeper, or several other places in the world. In terms of, are they negatively impacting the environment, there's still a lot of work to be done in the area to be able to definitively answer that yes or no. There's definitely aspects where we see the diseases that we see associated with honeybees also in native species. So the extent to which honeybees are potentially providing a conduit to vector disease in these native populations is a point of concern that people are following up to see how much of an impact that is. In New Zealand, particularly with Manuka honey, there's questions around taking lots of honeybees into these native forest blocks where there are a lot of native species, and a lot of native species, not only insects that are only found in New Zealand, and then how they are quite dependent on the nectar flow during this time of year as well. And then are honeybees consuming so much nectar? Because if we're seeing overstocking effects of honeybees and reduced honey production of honeybees because there's so many honeybees in the area, does that mean that we also are seeing reduced nectar availability for native insects as well? It kind of like the overstocking question, it's really complicated to tease out a definitive answer in that space. So it's hard to say one way or another yet. And then we also had a graduate student who completed her master's 2 years ago with our team at Plant and Food, and she was looking at myrtle rust as well, so a plant pathogen, and that honeybees will go actively forage myrtle rust. They'll pack that on their corbiculae, they'll bring it back to their hive, and you can actually feed even larval honeybees myrtle rust, and they'll develop normally. And so it is actually a protein source for honeybee colonies as well. And it actually, when the chemical analysis of it, was a pretty good protein source in terms of the amino acids that were available in it. So there's still the question of, does it get back from the hive back out into the environment, particularly if we're moving colonies to chase Manuka honey from Northland down to areas that maybe don't have as much myrtle rust in their bush blocks. But still, again, hypotheses that were being followed up, but we're not to a state with the understanding yet to say definitively one way, yes or no.
Right.
A lot of it depends answers, which are never as much fun as just being told what it is.
Yeah. We're all aware of the effects of our environment on bees and how they're impacted by pollution and chemicals. We touched on this earlier, but I read that by far and away the greatest amount of residual chemical in a hive are miticides. What have your studies shown about their toxicity to bees?
Yeah, so I have done less directly on miticide toxicity around bees myself. Definitely miticide residues are what's going to be the most common residue that you find into a colony. The synthetic miticides that we used have gone through quite a bit of testing to determine that they're of low toxicity to bees, but that doesn't guarantee that it's none when it's a chronic exposure at an incredibly low dose. And maybe it's not a direct toxicity like mortality, but does it have flow-on effects with just the kind of vigor of the individual bees or the colony? Fortunately, residues that are contained in the wax often stay in the wax because they're quite lipophilic, and so they go in and get trapped into the wax comb itself. But there's the question about brood that are just being reared in those combs. And there's also been work done with queen cells that were laced with various different pesticides, not only miticides, and some of the sublethal effects that are potentially seen. Yeah, still, I mean, another friend of mine who does a bit more research directly in this area likens miticides to kind of chemotherapy, where sure, we know that they're not ideal. We wouldn't just give them to the bees if we didn't need to, but varroa is much worse. And so we do need to deal with that. So kind of that balance of what risk is better to take and for ensuring varroa control.
This time of year, our thoughts turn to wax moths and how to protect our hives from them. Do you have any advice from your research?
Yeah. Wax moths is such a tricky one. And something that we didn't have as much issue with in the way that we managed our combs in Florida. So been a bit of a learning curve for me as well coming here. We often have just had open sheds where we've been able to crisscross frames, or we actually had large -20s. So I mean, I've only dealt personally with an apiary that was running at a scale of 100 to 200 colonies at work, not my own ones. We said that was like 1 to 15. But I've always been somewhere that's had a large walk-in freezer. So we maintain important combs in the freezer because it's just the place where you're not going to have a wax moth problem. And anything that's out that's black comb, wax moths are going to be quite keen on. Honey super boxes, if you can have them where there's airflow, so in a shed that they can crisscross stack, helps to minimize that. But we just had to transfer our whole operation for work into a shipping container because all of our storage into a shipping container. We still have our -20, but that's the shared space. So we don't have too much. It's in the freezer. We've had an open shed that we've been able to store in, but now we've shifted into the shipping container, which is pretty common with a lot of the commercial operators that we've gone around and worked with out in the field. And the wax moths are just full on in that shipping container. So I guess don't have a great amount of recommendation in terms of some sort of like fumigation protocol or something like that. We were wondering if something along the lines of the heating that gets done similarly with Onozema was something that we could follow up and try to see if that had an impact on the wax moth eggs and if that would have any benefit for beekeepers, because we haven't had funding to do that yet. So on a hobbyist scale, if you have a chest freezer and you have a couple hives and you can just put your frames in the freezer, that's great. It gets a bit dicey on a large scale to have that much freezer space.
Interesting. To me, one of the biggest mysteries in beekeeping is drone congregation areas. In New Zealand, we're fortunate to have 2 naturalized Kiwis that I know of who study DCAs. You were looking into varroa infestation of drones. Was this to see if it was a significant vector in the spread of varroa?
Yeah, I'm super excited to get to start doing some more DCA work here. It was a part of my master's project was doing DCA work, and then I kind of got away from it for, it's probably been about 7 years now. So I was quite excited to get a bit of funding that we could work on that with. And it was during my master's collections of drones that we had a bunch left over in the freezer as I was preparing to move to New Zealand. And someone had mentioned, well, we talk about maybe there's drones at DCAs, but we haven't, or maybe there's Varroa at DCAs at these mating sites, but we don't know for sure. Maybe that's a vector for spread, but we don't even know if they're there. I was like, well, I have a bunch of drones in the freezer left over from my mating sites that I sampled. I'm going to have to throw them away because I'm leaving the university and no one wants to maintain all of my tubes of samples in the -80 because it's valuable space in there. Let's just do mite washes and see if there's Varroa on there. So we were able to recover Varroa and actually recover different concentrations of Varroa from the sites that the mating sites that we collected out in the feral population compared to the mating sites that were nearby managed apiary sites. Suggesting that the mite control that you're seeing on the ground in the managed sites is limiting what you're seeing up at the DCAs and those mating sites on the drones as well. So we published that, and that's kind of the state of the literature on Varroa at mating sites, is that, hey, they can be there. So we have another master's student with the University of Waikato right now, who she did a summer project as a summer undergraduate last year and sampled 6 DCAs to see if there was Varroa at those mating sites. And then we also had mites per 300 bees numbers for the apiary that was on the ground nearby. So there's lots of drones from other places that are also ending up at that mating site, but at least that nearby apiary is representing a significant contribution to that site. And we saw evidence to suggest that you probably can see a correlation between what the mite infestation rate is on the ground and what the mite infestation rate is up in the air at that mating site. So she's following up with that more and has sampled 10 more mating sites and apiaries throughout the Waikato this summer. And we're adding that to the data set. It's quite a complicated relationship, so it's going to take a lot more than 16. I think she had 6 the first year, maybe it was 5. Going to take a lot more than even 20. So less than we have to definitively define that relationship. But we're hoping to get to the point where we would be able to have the mating site samples of drones and then say, oh, there's actually a really high risk for Varroa infestation for colonies in this area. So about a 1/4 kilometer radius around the mating site is where you expect those drones to be coming from their home colony that they're coming from. It can be up to 2 kilometers, but they don't like to fly very far to the mating site. So as a way to assess the regional risk of mite pressure in your area, so it's not just you testing your own colonies or a large-scale apiary beekeeper, it's not you having to test 100 colonies in your apiary. You can test at your mating site and understand what that looks like for the Varroa pressure. I think the biggest risk for transmission of Varroa from DCAs is that drones are actually quite prone to drift. So they don't always go back to their own hive. They often go back to a different hive. They're just not as precise with where they're going and paying attention to directions as the workers are. So that risk, if you're in an area, if it has a really high Varroa pressure and you have lots of drones with Varroa at your mating sites, then you're likely to see a lot more drift, particularly from those drones as well. And just understanding those dynamics and how they contribute to our management. We often don't think about drones, or we think about trying to reduce the number of drones that our colonies are making because they're wasting space with drone comb. So really starting to think about this other element of the colony and how that contributes to what we're seeing in our management strategies.
In the American Beekeepers Journal, there has been a study by Randy Oliver on pollen patties, which is reviewed in this edition of our journal. This looks at the efficacy of different brands and natural versus substitute. You also studied protein feeding. What was the emphasis of your study?
Yeah, so we similarly had 4, it's been a few years now, I don't remember the total number of different brands of commercially available protein patties. So we fed those out to colonies over several weeks and then tracked from the before feeding to after feeding, and then 8 and 16 weeks after feeding, what the colony dynamics or profile looked like. So we mostly looked at the number of bees and the amount of brood that was in the colony. So looked at colony size and colony strength through that time period. It was in the early, oh my gosh, I can't remember seasons anymore because I've gone from the northern and the southern hemisphere. So it went from August, into September, October. So it was late summer into autumn. There we go. Yes. So it was into summer into early autumn feeding. So it wasn't a spring buildup.
That would have been, sorry to interrupt, that would have been spring, would it? August.
No, so that's why it's so trippy, because that is the end of summer in the northern hemisphere.
Oh,
so
right. So
it was
sorry.
Yeah, this is why I've just lost all ability to identify what season it is. So it was late summer into early autumn that we were following the colonies. And in Florida, that's a common time for beekeepers to supplementally feed, because there happens to be a bit of a dearth in what's being produced for the flowers. There aren't a lot of flowers that are in bloom, so there's not a lot of pollen available during that time period. We didn't see really clear impacts of feeding versus not feeding in our treatments, but there seems to be a lot of that type of unpredictability about the benefit that you'll see from feeding protein in the literature. So there were some other trials that we read about and cited in the discussion of that trial, where they, in 1 year, would have fed pollen and seen an increase in honey production. And then the next year fed, protein patty. And then the next year they would have fed a protein patty and then not seen an impact. So there seems to be quite a lot that we haven't identified as the factors that determine if a protein patty is going to have a benefit or not. There isn't great evidence that it has, or there isn't evidence that it has a negative impact. For the most part, we do in areas like Florida that have small hive beetles. If you're just putting heaps of pollen patty in there and they're not able to consume it all, then small hive beetles actually are like, we'll eat this, thank you, and do quite well in the colony. So you do have to be careful with other pests that are associated with it. But there's environmental conditions in which protein feeding can impact positively the colony and increase productivity of honey downstream. Mm-hmm. But there hasn't been a clear definition of what those environmental conditions are and how you can tell if this is the time when it will versus the time that it won't have a positive impact on your profit in your apiary.
Did your study touch on nosema or dysentery when you were studying the protein?
Yeah, so we looked at the spore counts of nosema. So we weren't identifying if it was Apis or Serrani, but Serrani is the most common subspecies of nosema infestation that you see in the United States now. It's almost completely replaced Apis in terms of samples, but we did the spore counts where you're crushing a sample of bees and then looking at the number of spores that were held in their gut contents. We did that because in a laboratory trial, we had fed nosema to bees and then also fed different protein supplements and then saw an increase in nosema when you were feeding the pollen patties versus not feeding protein, which is a pretty harsh control because they're going to be eating something in the apiary side. But that protein source is what the nosema is also feeding off of in the gut. So they need that in the gut for them to be able to reproduce, or for the disease to be able to reproduce quite well. So when we didn't have a protein source available, there wasn't nosema buildup in the laboratory study. But when there was the protein patties available, and there were 4 different brands, we saw that increase in nosema. So we looked for that in the apiary, but the negative control is not being fed a protein patty, but still has pollen in the colony. So we didn't really see a difference in the nosema buildup between the treatments.
And lastly, what's your favorite book on bees or beekeeping?
Oh, yeah, there's a few. I guess from more of an academic standpoint, the book that I refer to most often when I'm exploring any new idea about bee biology is the Honeybee Ecology by Tom Seeley. It's actually quite a bit of an older book, but it's just a really nice, thorough discussion of the state of the knowledge in the 90s, probably, of basic honeybee biology, physiology. There's certainly work that's gone on since then to refresh on, but it's a really great foundation and has a really wide array from what's the temperature range that an individual bee can forage at and what happens if it's too hot or too cold while it's out flying, all the way to talking about swarming and collective behaviors in the colony as the colony as a whole. So it's a really, yeah, I go back to it all the time. So I really enjoy that book quite a lot. Less of a casual read, but actually really accessible the way that it's written. It's actually a really great little book.
Excellent. Well, thank you very much, Ashley, for sharing some of your studies and research, and it was very interesting. Thank you.
Oh, beautiful. Thanks so much for having me, Ken.